Assalamualaikum and a very good day. Today we are going to present a research article regarding integrating intracellular and extracellular proteomics profiling for in-depth investigations of cellular communication in a model of prostate cancer. As for the introduction, the research is fundamentally a cellular-based proteomics approach since the core methodology involves integrating both intracellular and extracellular proteomics profiling. The research also provides an understanding about cellular interaction and communications via protein because it emphasizes that disruption in these processes can contribute to disease development and progression. It also enhances understanding about cellular communication networks in prostate cancer development and progression, especially regarding how signaling networks contribute to disease progression and metastasis in castration-resistant cancer. The development workflow is not only limited to a proteomic aspect, but also it can be expanded as metabolomics and lipidomics studies, as it can be integrated for a multi-omics workflow. Lastly, the research provides a more complex picture and a better understanding of disease and their impact on multiple systems. It offers a holistic view of cellular mechanisms by understanding how proteins communicate and modulate each other. There are three objectives that can be achieved from this research. The main objective is to develop and validate a workflow for simultaneous profiling of intracellular and extracellular proteins. Secondly, it used this workflow to investigate cellular communication networks in a castration-resistant prostate cancer, also known as CRPC model. And third, to provide a reproducible and adaptable method that can be expanded for multi-omics application, including potential drugs screening. The methodology applied consists of four methods. First, cell culture, which is CRPC cells were cultured in a media containing TMSO and insulin transferrin selenium. Second, sample preparation, which extracellular protein is collected from conditioned media and precipitated with acetone digested with trypsin. For intracellular proteins, it is a harvested cells which is lysed and digested the similar method with extracellular proteins. The peptides is analyzed using high-resolution mass spectrometry, and the identifications and quantifications of peptides is performed using Proteome Discoverer. To analyze the data, it uses functional annotation via DAVIC and Cytoscape, the statistical analysis with Perseus, and R enrichment analysis with Metascape. This is the simplified workflow diagram to summarize the processes involved in this research. To sum up the main result, the study identified over 8,000 proteins with consistent coverage across sample groups, which demonstrating a high reproducibility. And most proteins were shared among extracellular and intracellular fractions. PCA confirmed sample robustness, while GO enrichment validated the extracellular protein captured. 93 out of 142 unique extracellular proteins remain after filtering, with pathway analysis highlighting cell morphogenesis, TGF-beta signaling, and ECM organization. Androgen receptor and PCA were absent, aligning with the CRPC models, but TGF-beta1 was highly expressed, suggesting it as a potential therapeutic target for advanced prostate cancer. The conclusion that can be made from the research is this research demonstrates that this robust and flexible workflow enables simultaneous analysis of both intracellular and extracellular proteins without loss of proteome coverage. This supporting the integrations and metabolomic and lipidomic studies for future multi-omics integration. The complementary data provide deeper insights into cellular communication and modulation to generate new hypotheses about the role of extracellular proteins in tumor phosphorylation. Overall, this method enhances understanding of cellular mechanism in disease progression, especially in aggressive prostate cancer models like CRPC.
Assalamualaikum. My name is Nurana Natasha, and I will continue with the principle of method chosen. In this research, the team used an integrated proteomic approach to study prostate cancer cell in depth. They extracted both intracellular and extracellular proteins to get a full picture of how the cells communicate and interact. Using a bottom-up proteomics method, the proteins were digested into peptides and analyzed with LC-MS for detailed identification and quantification. Finally, bioinformatics tools like gene ontology and pathway analysis help map this protein to biological pathway, showing how they are involved in processes like cell signaling, tissue remodeling, and cancer progression. To continue with, the result of this study gives us new insight into how aggressive prostate cancers behave, especially the types that resist treatment. By combining data from both inside and outside the cells, researchers can develop new biomarkers and possible targets for therapy. This holistic approach supports future multi-omics studies, drug testing, and personalized treatment, making it an important step towards better care and outcomes for patients with prostate cancer.
Hi, Assalamualaikum. My name is Nur Syafiqah Alamin Farid. So today I will present about advantages and disadvantages of study integrating intracellular and extracellular proteomic profiling in model of prostate cancer. First advantages is holistic approach, where both intracellular and extracellular proteomic profiling providing a more comprehensive picture of cellular communication than study that focus on one compartment. Next, reproducibility and scalability, where the highly reproducible across biological replicates and adaptable for highly throughput study and multi-omics integration such as metabolomics and lipidomics. The last one is relevant to clinical model. The use of unique prostate cancer model such as CRPC and AR negative allows the study of emerging cancer phenotypes that are poorly understood, thus offering new understanding. Okay, next, moving on to the disadvantages of integrating intercellular and extracellular proteomic profiling in model of prostate cancer, where first one is potential contamination in extracellular fraction, where the highly overlap between intra and extracellular protein identification suggests possible contamination from live cell, affecting the reliability of purely secreted protein identification. Second, limited to in vitro cell model. This method is limited to cell culture models. This restricts its direct clinically applicability and overlooks complex tissue level interaction. And last one is no validation via external data set. This study lacks direct validation or comparison with other published dataset or clinical sample to certain findings. And that's all from me, disadvantages and advantages.